Hey, welcome back to this screencast where we're talking about the cell-mediated immunity, and we went down the center pathway here. Now I want to continue and go down this right side over here and talk about what's going on with this aspect of the cell-mediated part of this. So similar to going down the center, now we're going to have like this infected cell here that has engulfed this antigen fragment, or it not really engulfed, but the pathogen actually directly infected the cell. And so this cell is in some way metabolizing this and will be triggered to express genes that then make an MHC class 1, again, so major histocompatibility complex 1. And again, the antigen binds with this MHC, and then this infected cell presents. So now this is a presenting act right here. It's presenting the antigen to the immune system and saying like, hey, look at me, I'm infected over here. And when this presentation happens, you now get a cytotoxic T cell that will actually bind. And so this T cell receptor here binds. And again, we know that when things bind together, you get a conformational change, and then you're going to get some sort of response on the part of this cytotoxic T cell. So let's look at that in a little bit more detail here. So going from left to right, here we have our infected cell over here, and it is presenting the antigen here with MHC class 1 proteins here and here. You now have this cytotoxic T cell that has a CD8 receptor thing going on here, so the CD8 receptor, and you bind that MHC class 1 there. That binding is then going to activate this cytotoxic T cell. And so what happens is the response is that this cytotoxic T cell in this example releases these little molecules here, and these are enzymes that are called perforin. Here they are, perforin as in perforate, as in punch a bunch of little holes in this cell. So these porphyrin molecules will create little perfs, little holes, you could also think of it as pores here. And what then happens is ions will diffuse in. That changes the water potential of this cell, makes the water potential a lot lower than the surrounding fluid, and then water will osmose in, and water continues to osmose in, and what you then have is this infected cell rupturing right here and lysing. Right? And so this infected cell is now no longer infected, maybe won't be producing more viruses, right? And you then curtail the effects of that infected cell or that cancer cell in the process, okay? So this is the cytotoxic T cells. You can see how that when these T lymphocytes here bind to an infected cell, how indeed that is toxic to these cells here, right? Let me get a little crossbones here as well. This is the bone. These are crossbones. That's awful. Anyway, so cytotoxic T cells binding. Okay. I'm going to introduce HIV here just for a second. This is a virus, a pathogen, and we're in previous screencasts we looked at how viruses are functioning, right? And so here's HIV binding and entering its host cell. Well, I will tell you now that this host cell are helper T cells. And so HIV infects these helper T cells, amongst other cells, but it will infect these helper T cells and actually cause ultimately these helper T cells to die. Right? So this is actually a sad state for these helper T cells. And as you can imagine now, whoops, if we go back to this diagram here, if these helper T cells are dying, well, then this whole thing is not going to happen, right? You're not going to get memory T cells, you're not going to stimulate cytotoxic T cells, etc., and your immune system then crashes, right? And so what you end up having then, this virus is called, the HIV is called human immunodeficiency virus. Immunodeficiency immunodeficiency, right? Your immune system is deficient because the central cell, that helper T cell, is being actually destroyed by these viruses, okay? So that's a problem. All right, so by the end of these two screencasts now, 2A and 2B, you should be able to describe and explain cell-mediated responses involving the helper T cells, the cytotoxic T cells, antigen-presenting cells, and a little bit about these MHCs, what they are. And we did not talk about memory T cells and memory cytotoxic T cells and helper T cells, but we'll do that in another screencast as well. Okay, thanks for listening. Bring your questions to class.